Welcome to the Polymicrobial session. Thanks for joining us this morning early. We have just a couple slides to orient you guys and kind of plant some ideas in your mind as you see the talks today. So I guess we will get started. Yeah, we're going to tag team a couple slides so as folks come in. All right.
So this session is focused on polymicrobial infections and interactions. And so that brings up a number of questions. So among those are, what are the ideal organisms and models that we use to study? So these can be really simple in vitro models, just throw in a couple organisms, some broth, maybe some interactions, and see what happens. There's more of an in situ, slightly more physiological, let's say in sputum. And you can also do these more basic ones in an environmental context, such as using artificial media or other specialized growth conditions.
And then, of course, like which model are you going to use? So, a mouse model versus ferret, rat, pig. And then also for polymicrobial, kind of drawing in from some of the sessions that we've already had, even the microbial ecology session yesterday, for example, we know that there's polymicrobial interactions where one microbe can influence the behavior of another microbe, but also one microbe could influence the behavior of the host, which then can influence the behavior of another microbe. So there's these complex, not just microbe-microbe interactions to think about for polymicrobial infections, but microbe-host and microbe environment, and this kind of interplay. And so this figure here was actually a summary from ECFS meeting, a polymicrobial session a couple years ago, and so we wrote a little review and kind of summarized at that time some of the polymicrobial interactions that folks had talked about. And so this slide even could be updated after today and after this meeting. Okay.
And then finally, you have your model. You have your hypothesis. So what are your analyses? Of course, again, you can do some just classic growth bacteriology. You can do molecular genetics, which we hear a lot about now, such as the 16S or metagenomics or metabolomics, whole genome sequencing. You can actually look at the interactions, which is an important thing to do. Being scientists, observation is key. And of course, the big thing now has been the host and bacterial or pathogen metabolites. So, and again, we'll hear about all of these different methods. So our first speaker is Phoebe Shi, and she will be talking about fungal and bacterial competition.
Thank you so much for coming this early. And my name is Phoebe, and I'm currently a postdoc fellow working with Harmeet Malik and Ajay Dentika in Seattle. And today I'm going to tell you a story about the basic science of fungal bacteria competition. It is not very clinical relevant at this stage, but I hope this story will bring us some new perspective to think about polymicrobial infections. This is my disclaimer slides. And in general, I'm very interested in understanding how fungi and bacteria interact. Both of them, they can form polymicrobial communities either surrounding us or on us. In those environments, they can either cooperate with each other or they can compete with each other. But however, up to today, we actually know very little how they actually interact at the molecular level. I think the big issue is there are just so much different strains of microbes exist, and then they are exist in different variety of environment. That makes this question challenging. But however, I want to argue that understanding these fundamental aspects of their interaction is critical because this will allow us to engineer microbial community even for either the medical or agricultural purpose. So during my postdoc, I specifically focus on two species. One is the Candida albicans, the fungus, and then the other is the bacterium Pseudomonas aeruginosa. Those two opportunistic pathogens, they are commonly found in a variety of polymicrobial infections in humans, such as in cystic fibrosis lungs. You can also find their coexistence in the chronic wounds. And the most annoying thing is when they coexist, they change the development of the disease progression. They can allow the microbes become very drug resistant, and therefore treating this polymicrobial infectious disease becomes very hard. And that is why it is very important for us to understand how they actually interact. In the past, a lot of study has focused on how Pseudomonas interact with the Candida albicans. And we know that the bacterium has a lot of strategies to suppress the growth of the fungus. However, there's one question we actually do not know, which is if these two species can coexist, how does the fungi compete with the bacterium in this context? So during my postdoc, I identified a condition that we actually see the fungus can suppress the growth of the bacterium. So I decided to use this condition to address this question. But in order to address this question, instead of focusing on the fungal side, I actually decided to focus on the bacteria side. And I'm going to tell you why we decided to do so. So we reason that, if we see the fungus can suppress the bacterium, then that means the bacteria must use some defense strategy to protect itself in this condition. And such defense strategies can be encoded by some defense genes. So as long as we can find a way to identify those defense genes, we will be able to understand how does the fungus suppress the growth of the bacteria. So that's how we were thinking about this. So how are we going to identify those defense genes? As a geneticist, I think if we can identify any mutants in the defense genes, then we will expect those mutants will grow very worse in co-culture with the fungus. So to identify this defense gene in a more systematic manner, we use the transposon sequencing on the bacteria side, try to identify all possible defense gene mutant we can find. So I used this approach, and then we finally nailed down to 3 gene candidates. These are the genes from PA4826 to PA4824. These 3 genes, they are right next to each other, and we suspect they are in one single gene operon. Interestingly, their expression is induced when they are in bacteria in co-culture with the fungus. So what do those genes do? We do not know the function of the 26 and 24, but we do know the middle gene, the 4825, encodes a magnesium transporter known as MGTA. This gene is induced in the low magnesium condition. So when we see this, we were pretty excited, and then we decided to verify our screen result by creating the single gene deletion and test their effect in co-culture. What we found is, in the end, we were able to identify and confirm that PA4825, this magnesium transporter, and its downstream gene, PA4824, are required for the defense against the fungus. Through several experiments I'm not going to talk about here, we are also be able to identify that in addition to 4825 originally known involving magnesium transport, 4824 is also very important for magnesium uptake in the bacteria. So we conclude that, at least in our model system, we found these 2 species compete for magnesium. This is kind of exciting because this allows us to see a new axis of competition between these 2 species. And then we start to wonder, is this mechanism general or very specific to these 2 species? Using a panel of different fungal species and bacterial species, we actually found this competition is a very general. We can see a lot of Candida species, even Saccharomyces cerevisiae, can sequester magnesium from the bacteria and suppress the growth of several gram-negative bacterial species. And this is kind of cool because in the past, people have been thinking about how iron as a very important player for nutritional competition. But this is the first time we see magnesium can be another axis of nutritional competition. And I started to wonder, why should we care about this? How does a magnesium competition affect the bacteria? I want to address these questions both from the bacterial physiology and also thinking about how magnesium competition can affect bacterial evolution. And before I show you some data about what we have known so far, I want to remind you, magnesium is a very important cation inside the cells. It involves in multiple essential cellular processes from the function of ATP to the protein translation. But in the rest of my talk, I'm going to tell you about what we found about the role of magnesium on the bacterial outer membrane, because outer membrane is a very important target for several antibiotics. Just a brief summary about what the bacterial outer membrane is. So bacterial outer membrane is a very spatial structure for gram-negative bacteria. It is composed with a the double lipid bilayer, and also there's a negatively charged lipopolysaccharide on top of that. Since this is a very specific structure in gram-negative bacteria, it is often used as a target to suppress the gram-negative bacteria just between the interbacterial competition. For instance, a molecule produced by gram-positive bacteria, now we know as a colistin, this molecule can actually target the LPS bound by the magnesium. So in the normal situation of magnesium condition, magnesium will attach to the negatively charged LPS, therefore maintain the membrane integrity. But however, this structure is actually bound, can be targeted by this drug. And therefore, in this condition, that will lead to the cell death if the gram-negative bacteria sees colistin. And that's why colistin has been used as a last resort of antibiotic to treat a lot of multi-drug bacterial infection in the clinic. However, in the low magnesium condition, bacteria actually will modify its LPS and prevent the binding of the colistin. Therefore, in this condition, bacteria become resistant. Knowing that the low magnesium can induce bacterial resistance to colistin, we wonder what happens when fungi sequester magnesium from the environment. Does it also impact the colistin resistance on bacteria? So we set up some antibiotic survival assay. We want to assay what is the survival rate of Pseudomonas aeruginosa in monoculture versus co-culture. This is what we found when Pseudomonas grow in monoculture. We know the bacteria are very sensitive. The survival rate to a range of colistin concentration is around like below 10%. But when the bacteria in co-culture with the fungus, the survival rate drastically increased. This suggests that the fungus indeed can protect the bacteria from colistin, and suggesting that this competition actually provides a benefit in an antibiotic-containing condition. And this is kind of exciting because it tells us that the fungus actually changed the bacterial physiology. But if we think about the polymicrobial community, these two species, they can live together for a while. And I'm very curious, how does fungus in this context can affect the evolution of colistin resistance in bacteria? So to address this, I use experimental evolution. I want to evolve bacteria to become colistin resistant, but in two different conditions. So basically what we did is we take the wild type PL01 population, we passage them in a condition with the colistin, either in the monoculture condition or in the co-culture condition. Along the course of evolution, we increase the concentration of colistin from 1.5 microgram per ml to 192 microgram per ml to select for colistin resistant bacteria. At the end of evolution, we analyze our final evolved populations across these two conditions and see what's the difference. So we know that after evolutions, both my replicate population in both conditions, they can become very colistin resistant. Here I show you the survival rate of bacteria to the highest concentration I use in my experiment. And I want to remind you that my ancestor actually cannot survive in this context. So the fact that many of the population survival rate can reach more than 50% is pretty strong phenotype. After that, we did some whole genome sequencing, try to understand what are the evolved mutations in those conditions. In short, we found that populations in these two conditions, they actually require different genetic roots to become resistant. We noticed that our monoculture populations, they become mutators because we found each population, they had at least hundreds of mutations. And this is not insane in the co-culture conditions. When we look into the mutations more detailedly, we found something interesting, and I'm going to show that in the next slide. Here I show you the genes has been mutated across multiple replicate population, here first in the monoculture conditions only, and I show you that to specifically highlight that the known genes for colistin resistance. So what we noticed that a lot of our monoculture populations have mutations in the genes already known for colistin resistance. I want to highlight that we see a lot of mutations in genes involving DNA repair. That explains why we see those populations become mutated. We also see mutation in the two-component system pathway, PMRB. This is a known genes involving colistin resistance. But we actually rarely see those genes become mutated in co-culture condition. Instead, co-culture populations, they require different set of mutations. We found a lot of mutations occur in one of the LPS synthesis pathway gene, HTRB2, and to our surprise, the other putative magnesium transporter I identified previously actually mutate across all our 8 replicate population in co-culture conditions. So this is a very interesting, and then we have been thinking a lot about why these co-culture populations require so few mutations, but they can become equally resistant as the monoculture populations. And we reason that maybe after evolution, this evolved resistance in co-culture could be still highly depends on the presence of Candida albicans. So to test this hypothesis, we went back to our culture evolved population and see what happens if we remove the fungus first, and then the test colistin resistance. If my hypothesis is right, I will expect that colistin resistance will be reduced. We can use the same setup to ask, is this resistance depending on the magnesium level by adding magnesium in my co-culture condition? And this is what we found. So for the first population, population number 1, this is the result I show you. Before fungal removal, evolved strains are very resistant. When we remove the fungus, we notice that the survival rate significantly decreased, as well as when we add the magnesium into that condition. This phenotype is not just specific to population number 1, it is also true across all my replicate evolved population. So this tells us that this evolved resistance does still depend on the fungus or the low magnesium condition. So this evolution experiment does tell us that the competing fungus actually can have an impact on the evolution of colistin resistance. What I've shown you that is they can evolve through a non-canonical mutations. Our current work actually shows that these mutations can dramatically change the LPS structure. As a result, even though the cells are resistant to colistin, but they have pretty bad defect on their outer membrane. So in the conditions without colistin, these cells actually have very strong fitness codes, and they grow very badly in conditions without colistin. Although all these findings are cool, but to me, the most exciting questions this evolution experiment actually reveal is, why does evolution occur this way? What I mean is that, why are these mutations specifically selected in co-culture conditions, but not in the monoculture conditions? So we are currently testing an idea that maybe it is due to the environment, the magnesium depletion created by the fungus. This environment might be able to change the membrane modification on bacteria, therefore deciding what kind of mutation can be selected in this context. So in this talk, I have shown you that by focusing on the Candida albicans and Pseudomonas aeruginosa, we discovered 2 magnesium transporters are required for bacterial fitness in this context. This allows us to find that magnesium competition can be a general and novel form of fungal bacteria competition. Even though this is very general, but this competition does not necessarily only bring the deleterious effect to the bacteria. In condition with the colistin, this competition actually can protect the bacteria from antibiotic treatment. We also found that this magnesium sequestration fundamentally alters how bacteria evolve antibiotic resistance. We see a lot of mutations that have been reported previously are involved in this resistance. And after evolution, this evolved resistance still depends on the fungus. So we also found that in this condition, if we remove the fungus, that can make the resistant bacteria become very sensitive to colistin again. And this, like a very simple finding, and can actually have some biomedical implications and tell us to think about in a polymicrobial setting, when we want to treat the bacteria, maybe we need to think about what are their neighbors are and how do their neighbors affect their physiology. So with that, I want to thank people who have helped me to launch these research programs. I have 2 very amazing and supportive mentors, Harmeet Malik and Ajay Dandekar, and I thank them to allow me to start this new research program in both of their labs. And this science cannot be succeed without a lot of work from my mentees, Wendy Sung, and also very great staff scientist Jenna Young and also Nicole Smalley from both labs. And I learned a lot about Pseudomonas biology through collaboration with Deb Horgan and also Robert Ernst at the University of Maryland. And in the end, I want to thank my funding resource, especially special shout out to Cystic Fibrosis Foundation. I thank them for supporting my current and also the future research. And thank you for your attention.
Hi, I'm Katie Heiser from National Jewish Health. That was really fascinating and as a clinician, every time there was another talk yesterday about Candida and Pseudomonas, and it's kind of terrifying because we usually ignore Candida in the CF airway, and now I'm thinking we need to get rid of it because it's keeping Pseudomonas around. In terms of how you think the mechanism of the fungal dependent resistance to colistin, do you think there's a physical interaction happening between the organisms, or do you think, what is your idea for how the fungus, when you get rid of the fungus, it's now colistin sensitive again, so.
Yeah, so our idea, so this need to be tested. We think it's less related to physical interaction. It's more related to, because the fungus change the environment, specifically in this case, reduce the magnesium level in the environment. And as a result, bacteria will activate a pathway to modify the membrane. And that automatically will give some resistance to bacteria. But the fact that this new environment created by the fungus, we think can actually channel how the evolution could happen. And a lot of mutations this is my current hypothesis that we identify so far, I feel it is highly rely on the first step of modifying the membrane.
But so the mutations, though, in Pseudomonas that you found, those would persist even after Candida is
removed,
yes.
Yes.
But then it becomes sensitive to colistin again once the the fungus is removed?
Yes. So that means the effect of mutation does still require on the fungus is present. And then we currently are trying to do more genetic experiments to really figure out what are the genetic underpinnings of this like at the molecular level. And if we can nail that down, and I think this hypothesis will be more complete.
Thank you.
Yeah.
Hi, great talk. My name is Michaela. I'm from Imperial College. I just had a question about the magnesium concentrations that you used.
Yeah.
Were they at all similar to what the SCFM concentrations are, or is that what the?
Yeah, that's a really great question. So in summary, the magnesium concentration in this condition we use is definitely a slightly low magnesium medium. But because of simplicity of this system, we were able to nail down at what range of magnesium concentration we can see this competition. So in short, we know as long as magnesium concentration below, say, 0.5 millimolar, we start to see this competition. And I think in SCFM, for the media itself, the magnesium level is slightly higher than that. But in a lot of sputum samples from literature research, we found that there's a huge range of magnesium concentration. And the level what we found actually is within the physiological range that has been reported before.
Great, thank you, great talk.
Hi.
Hi.
Thanks for a great talk. I was just curious if you had thought at all about the possibility that magnesium starvation might be having really profound effects on ribosome biogenesis and ATP homeostasis, as has previously been described.
I think
Probably.
I definitely think that will. And also I think in Salmonella, there are a lot of studies focusing on how magnesium changes the translation in bacteria. And we haven't really tested about that, but I think that would be another great venue to explore. Yeah.
Great, thanks.
Hi. Hi, Barbara Karl from Münster, Germany. Very nice work, very interesting. So did you have the chance, because there are so many Pseudomonas aeruginosa genomes around, to look if this mutation that you found is also present in the published Pseudomonas aeruginosa genomes?
You mean for all the eval mutation we found, or for the 4824 mutation?
The MGRT?
The 4824 mutation. Yeah, I did a very quick search, and what I found is, at least from the strains I can download from pseudomonas.com, I don't see that mutations occur in the isolates, and that position apparently been very conserved across many Pseudomonas species.
Okay, and maybe a short other question. So did you also have the opportunity to look at Candida, if Candida is affected somehow by this competition?
Yeah, that's something we are currently looking into that now.
Okay, thank you.
I have one more question. And then Katrina does too. I'll ask mine first. I was wondering, so your experimental evolution regime, was it done in planktonic culture? And if so, do you have any expectations or predictions of how the evolutionary trajectory might change if you instead evolve the two microbes in biofilms?
I think that probably definitely will be changed, because there could be, so in my experimental setup, I only select for the fitness of the cell. But if they are in biofilm, then there will be many different parameters that matter, not just the doubling time, like how fast the cell can grow. And I always expect we might be see more other types of mutations rather than the one we found. But there could be some overlap between the one we found and also in the biofilm setting.
Thank you.
Hi, I'm Katrina Whiteson from UC Irvine. And I wanted to ask you about if these mutations that you see in the co-culture context arise in nature. And I think you just said in the question from Munster that on pseudomonas.com, none of those genomes have the mutations that you found in the co-culture with Candida. That's really interesting. I mean, Pseudomonas and Candida are clearly together in the airways. So why do you understand that? Or?
Well, that's a good question. I have no good answer about why I don't see that in nature. Well, people can argue maybe we did not look harder, but but maybe there could be in nature, there might be other mechanisms rather than there could be like multiple ways these two species can interact, right? And what we identify might be just one of that, and we sort of like enrich the importance or priority of this interaction and see what is the effect of such competition impact on the bacteria evolution.
Maybe if you look in metagenomes, the mutations might exist.
That's a good idea, But I also haven't looked really completely. Yeah.
Thank you.
Okay. Thank you.
Thank you. All right. Next we have Kendall Schaefer.
All right. Hi, everybody. Thank you for being here this early. I'm Kendall Schaefer. I am a PhD student in the lab of Dr. Susan Burkett. And today I'm going to be talking about co-infection with Staph aureus and Pseudomonas aeruginosa. So I have nothing to disclose. So as I'm sure you're all familiar with, CF and respiratory infection really go hand in hand. And as you can see in this graph, Staphylococcus aureus in green is the most prevalent pathogen in the lungs of people with CF in the younger population. However, older patients with CF often have more Pseudomonas aeruginosa, and you can see that switch here. We don't know why this switch occurs, why older people with CF have more Pseudomonas rather than Staph, but we do know that co-infections occur throughout life with both Staph and Pseudomonas. So as I mentioned, Pseudomonas and Staph co-infection is extremely prevalent in people with CF. And you can see that here in this study by Fisher et al., in which they studied a longitudinal cohort. And at the beginning of the study, about 30.6% of people had Pseudomonas and Staph both isolated from the lung simultaneously. However, at the end of this 12-year study, that had increased to 50.7%, which indicates that either as people age, co-infection is becoming more prevalent, or simply with time, co-infection has become more prevalent. Regardless, this is indicating that this is a really important aspect of CF to study. So we also know that Staph colonization in early life is a risk factor for subsequently developing Pseudomonas infection later. Additionally, it is known that chronic co-infection is associated with worse clinical outcomes. But what's interesting is that in vitro, Pseudomonas is generally antagonistic to Staph. And we know that in the CF lung, though, they can coexist. So the mechanism there is not really known. Currently, most studies regarding co-infection with these two organisms have been done either in murine models or in vitro models. And these are great, but they unfortunately don't allow us to really look at chronicity very easily. So my lab has a CF rat model. We have a few CF rat models, but the one I'm going to be talking about today is the CF rat model, or the CFTR knockout rat, which is simply a rat with a genetic knockout of CFTR. And these rats develop a lung disease phenotype that's characteristic of people with CF. What I really want to drive home here is that these rats can withstand chronic infection of both Staph and Pseudomonas all the way out to 28 days, which is certainly chronic. So you can see in red here, the knockout rats are able to maintain infection all the way out to 28 days while their litter mates, their wild type litter mates, either nearly clear or completely clear infection by 7 days. So these rats are not eradicating chronic infection, which is great. So we're now able to study chronicity in infection. So knowing that, we really wanted to understand how Staph and Pseudomonas behave together in the CF lung during chronic dual infection. So to recapitulate the sequence of infection seen in people with CF, we first infected our rats with Staph aureus at 10 to the 7th CFUs in agarose speeds. And the strain we used is SA0831, and this strain is a MRSA strain isolate from someone with CF. And we let that infection by itself go for 7 days. At the end of the 7 days, we then either just continued with just the single Staph infection, or we infected with Pseudomonas aeruginosa. The isolate we used is PAM5715. This is a mucoid strain of Pseudomonas also collected from the lungs of someone with CF. And then we let that infection go for another 21 days for a total of 28 days. So at the end of the 28 days, we homogenized and plated the lungs for CF use. And what we found is really interesting. So first, unsurprisingly, you do see a bit of a decrease in staph colonization when it's in a co-infection with Pseudomonas compared to when it's alone. That's not super surprising because, as I mentioned, Pseudomonas is antagonistic to staph. However, what we find really interesting is that in a co-infection with staph, Pseudomonas actually does better. We are able to recover higher CFUs compared to when it's in a single infection. Additionally, compared to uninfected knockout rats and Pseudomonas-infected knockout rats, in a co-infection with staph and Pseudomonas, the CF rat lung develops much more mucus plugging compared to with just Pseudomonas or uninfected. So this is leading us to believe that co-infection with staph is promoting Pseudomonas colonization somehow, likely through priming the environment to make it more suitable for Pseudomonas. To investigate this, we used metabolomic analysis, and we chose this method because we believe that staph is likely altering the nutrient environment in the lung. So an important thing to note is that for our samples, we're using bronchoalveolar lavage fluid from rats. We have to use the Bly-Dyer method of extraction. And this is not the standard method that would be used, but the reason that we have to use it for our samples is because our rats have a significant amount of protein in the bowel, and that protein is mucin. Because not only do they have the CF phenotype, but when they're infected, they're producing a lot of mucus. So that BALF is then subjected to LCMS. And the way that we collect that BALF is we infect our rats with our SA0831 staph strain for 7 days. And the reason we chose 7 days here is because if you remember in my original sequential dual infection, that is how long the staph was in the lungs by itself before Pseudomonas was introduced. So we really want to see what is going on in the lungs right as Pseudomonas is introduced. And what we found is that at baseline, CF and wild-type rat lung metabolomes are really not that different. You can see here in the PLSTA plots that there's a decent amount of overlap between these two groups. However, when they're infected, the CF and the wild-type rat lungs have a very different metabolome, which is indicated by the clear separation here of these two groups. And we think that the differences in the metabolome during infection might be contributing either to the chronicity of staph or to the establishment of Pseudomonas infection. So we, of course, then wanted to see what exactly was changing. So we used two metrics for this. We looked at the PLSTA VIP, or the PLSTA variance, importance, and projection. And these scores essentially are telling you how much a specific metabolite is contributing to the separation that I showed you on the previous slide. The second is a simple t-test. And while there's a lot of metabolites here, I really just want to draw your attention to homoserine and lactate, as these are the two that I'm going to be moving forward with in this talk. So both homoserine and lactate are increased in the CF rat lung compared to the wild-type lung during infection. And to go a little bit into these metabolites, lactate, or lactic acid, is a metabolite that's produced by both staph and the host during infection. It's also been shown that people with CF have higher levels of lactate in their sputum than a healthy individual. And as a result, this is an ingredient in CF sputum medium, or SCFM. Additionally, it's been shown that lactate can act as a carbon source for Pseudomonas and can actually improve its growth and biofilm formation. Homoserine is another metabolite. This metabolite is involved in the biosynthesis of both methionine and threonine. And this is important because methionine has been shown to promote pathogen proliferation as well as biofilm formation for multiple pathogens, including Pseudomonas aeruginosa. And additionally, it is important for threonine synthesis, and threonine is important for the synthesis of mucins, which of course play a huge role in the pathophysiology of CF and CF-related infections. So just to cover all of our bases, before we move forward with testing the metabolites, we wanted to ensure that the effect that we saw of staph on Pseudomonas burden wasn't just with that one strain of Pseudomonas. So we repeated our sequential dual infection using an environmental strain of Pseudomonas. And this is because when people get infected with Pseudomonas the first time, it's usually from the environment. So we repeated this infection where we infected rats with staph at day 0 for 7 days, followed by an infection with Pseudomonas at day 7. However, the Pseudomonas in this case was PA098, which was an environmental strain. At day 28, we once again homogenized and plated the lungs for CFUs. And we found that just like with the clinical isolate, staph is able to promote Pseudomonas burden in the lung in a co-infection. So now that we know that, we want to move forward with looking at the metabolites. So to test these metabolites in vitro and their effects on, or in vivo, and to test their effects on Pseudomonas establishment in the lung, we grew our Pseudomonas strain in SCFM, either with or without the normal 9.3 millimolars of lactate that's included in SCFM, or with or without 100 micromolars of homoserine. We did that for 24 hours. We then encapsulated the Pseudomonas in agarose beads and infected the rats with this. At the end of the infection, we homogenized and plated for CFUs. And what we found was that while lactate does slightly and non-significantly increase the colonization of this environmental Pseudomonas strain, it does not do so as well as homoserine, which actually does significantly increase the colonization of Pseudomonas after just a pre-exposure to it. And so this is indicating to us that homoserine is somehow altering Pseudomonas to promote its colonization even better than lactate can. And so just to summarize, chronic co-infection with staph increases the burden of both clinical and environmental strains of Pseudomonas in the CF rat lung. And the differences in the metabolome between the wild type and the CF lung appears to be primarily driven by staph. And this makes sense because when you're doing a study using human sputum or something like that, it's hard to control for not having staph infections. People are colonized so early. Additionally, lactate and homoserine are both increased in the CF rat lung following infection with staph. And finally, growth of environmental Pseudomonas in the presence of homoserine before infection actually promotes its burden in the CF rat lung, similarly or even better than lactate can. So moving forward, we really want to understand the mechanisms behind this, behind the availability of the key metabolites themselves, as well as their ability to allow Pseudomonas to adapt to the CF lung. To do this, we're using RNA sequencing and genome variant analysis of our Pseudomonas treated with our key metabolites, as well as Pseudomonas recovered from the lungs during co-infection. We also plan to assess the effects of delivering metabolites directly to the lung during infection to see what effect they have on the bacteria during continuous exposure, as opposed to just the pre-exposure. And finally, in the age of modulator therapies, we of course want to understand how CFTR modulators are altering these staph-induced changes in the lung, or if they are, and then their effects on subsequent co-infection with Pseudomonas. And with that, I just want to thank my lab, the Targeted Metabolomics and Proteomics Laboratory at UAB, and my committee members for helping me with my project. Thank you.
Nice talk, thank you.
Thank you.
We have published in 2018 a paper on the chronic co-infection of Pseudomonas and Staphylococcus aureus using clinical isolates, and we saw similar results. So an increase of the persistence in mouse model, clearly, of the persistence of Pseudomonas aeruginosa. And for this reason, I would like to ask you two things. First, if you tested different clinical isolates from the early stage of colonization to the late ones, and if you have seen any difference. And the second one, if you have looked at the immunopathology, in particular in the parenchyma, I don't know with the histopathology or whatever. Thank you.
Yeah, thank you. So we have other strains of clinical isolates of Pseudomonas that are non-mucoid and that are more acute. We haven't tested them in an actual sequential dual infection in that way, but we do plan to do that. However, we have tested them in vitro. We had tested their responses to the metabolites that we found, and we found they have differential effects depending on the strain, but a lot of them do behave very similarly to the clinical isolate that I showed. And then to answer your second question, we have samples ready to analyze for different cytokines. We plan on doing a Luminex assay and trying to see what cytokines are present when Pseudomonas hits the lung, so with just the staph infection, as well as what is present after chronic co-infection with both staph and Pseudomonas. We haven't looked at any immune cell We haven't done any immune cell counts or anything because we send off the whole bronchoalveolar lavage fluid for metabolomics, so we don't separate them. But that is something that I think would be really important to look at for sure, and we do plan on investigating immune mechanisms next.
I have a question for you. For the with the lactate stimulated Pseudomonas, what have you looked at gene changes in that, and could that be part of the
So that is
adaptation?
and what's next. We actually just sent off our samples for whole genome sequencing and seeing genetically what's different. So we should get that data back soon. But that's certainly our next step because we do want to understand what's different genetically. We don't know that right now.
Properties of in your experiments with the environmental Pseudomonas aeruginosa, you added additionally the lactate and homocysteine, right? Did you do this also without? And could you see similar effects? So just the environmental Pseudomonas aeruginosa, did you also see the increase in
Yes, so for the sequential dual infection where we infected first with staph and then with the Pseudomonas, that was not with homoserine or lactate. That was just with staph, and because we wanted to make sure that this isolate could be increased by staph. After that, when we treated the bacteria with homoserine or lactate, we did do that with and without homoserine and lactate as well. And we saw that, go back actually and show you. We saw that the non-treated did not have, we weren't able to recover as much from the lungs compared to the treated bacteria. So, yeah, that answers your question, hopefully. Yeah, thank you. Hi. Yeah. Mm-hmm. Yeah, so this was at the chronic time point, which 28 days in this case. Mm-hmm. So we have collected the samples for that. We have that in RNA later ready to go, ready to extract, but we haven't gotten to that point yet, but we absolutely are planning on doing that.
I have a question for you too. Yeah. Great talk, by the way.
Thank you.
So I'm interested in, you mentioned in response to one of the other questions that there are some strain-specific effects. And so in CF in CF, most of the Pseudomonas aeruginosa that can be isolated from the lungs is from clade 1, so kind of a PaO1-like Pseudomonas, but the major clade structure is clade 1, 2, 3, 2 being maybe represented by PA14. So I'm curious if you've looked at maybe clade 2 Pseudomonas and if they respond the same way as clade 1, and could that explain your strain-specific effects?
So we've looked at PaO1 itself. We have not looked at really any clade 2. We've looked at other clade 1. So I think a lot of the strain-specific things is just more, like the PaO1 itself actually responds differently than PAN5715, despite them being very genetically similar. Mm-hmm. But no, I think that's a great idea. We do have access to PA14, of course, and so I think that would be a great thing to consider, for sure. Thank you.
Okay, thanks. All right, I think...
Any more?
Thank you.
Our next speaker is Amanda Morris, and she comes to us from Valerie Waters' lab at the Hospital for Sick Children in Toronto.
Hello, everyone. My name is Amanda, and I'm a RA in the Waters lab at Sick Kids Hospital, and I'm going to talk about the spatial and temporal association of Pseudomonas aeruginosa and Staph aureus in CF sputum during pulmonary exacerbation. I have no disclosures. So from the last presentations, you guys are probably familiar with this report here showing that the two most common pathogens that are infecting the CF lung are Pseudomonas aeruginosa and Staph aureus, and these two bugs have a frequent co-infection rate somewhere between 20 and 50%. And in the younger stages of a CF patient, Staph aureus is the most common pathogen infecting the CF lung. But as these patients age into adulthood, Pseudomonas becomes more predominant. So it raises the question as to whether these two pathogens are aggressively competing for nutrients in space, or do they have some sort of a mutualistic or commensal-like relationship supporting growth and antimicrobial resistance. So in literature, there's definitely some inconsistencies between the studies. An increasing number of studies are showing that PA and SA co-infections are associated with worsening clinical outcomes in CF patients. However, there are also studies showing that there's no significant clinical difference between PA monoinfections compared to PA and SA co-infections. And then there's also studies showing that there's poor clinical outcomes in PA monoinfections independently of SA presence. There's clearly like a modeling disconnect. So despite the use of things like clinical isolates, artificial sputum, and antibiotic concentrations that are achievable in human lungs, there's disconnect between in vitro and in vivo modeling and inpatient results. And this has a lot to do, what we believe, is to the lack of polymicrobial interaction, and just there's no evidence of spatial and structural organization of the two pathogens within the same microbiome and the overall biogeography of infection. So the closest approach that we believe for addressing this disconnect is a technique coined MIPAC, short for microbial identification after passive clarity technique. And the way the approach works is that you collect the sputum from the patient, you embed it in a hydrogel mixture to form a gel and keep the structural integrity of that sputum. And then this allows you to clear the sputum in a SDS solution, and then following clearance, you can use probes specifically for fish, specifically for, in this case, PA and SA. And you can also use, at this point, at the end, an antibody that's specific for other exoproducts or exopolysaccharides within the sputum of interest. And what this technique produces are these really neat images of modeling polymicrobial infection directly in the sputum. So this is just an image from a previous study that I showed, just looked at the difference between persistent and eradicated infection, where in green is the Pseudomonas, and then PSL exopolysaccharides overlapping in magenta, and then DAPI is a counterstain just to show in blue other organisms that exist within the sputum. And then with replication, this is from another study, you can clearly see that you can model the infections and see differences depending on when you collected the sample. So utilizing this technique, the objective of the present study is to investigate the interaction between PA and SA, or the secreted SA product Staphylococcus protein A, which is SPA, within CF sputum from patients with chronic PA infection, with the hypothesis that sputum with PA, SA, or SPA co-infection will have greater PA abundance, aggregation, and spatial closeness, with increasing SA or SPA abundance during PECs due to antimicrobial tolerance mediated by interactions in polymicrobial communities. So this is a very longitudinal study. It's taking place across 3 sites, so Sick Kids in Toronto, St. Mike's also in Toronto, and McGill University in Quebec. And we have so far enrolled 40 patients, and we have collected 146 sputum samples from these patients. And we are following the 40 patients at consecutive time points, collecting it before a PECs and also after and during PECs. So we're using Dr. Lipuma's framework for the BETR framework to classify these samples. So B being baseline, E is day 0 of PECs treated with IV antibiotics, and then we have big T. We're collecting it at 7 days, 14 and 21 days of antibiotic treatment, and then 60 days as well. And then we also adapted this technique to add the lowercase e and lowercase r, and this is just to indicate PECs that's treated with oral antibiotics and recovered from there. So in terms of the project workflow, I'm looking specifically using MIPAC, but I just wanted to add that every time we get a sputum sample, a split portion of it goes towards other analyses. So we do real-time PCR to determine PA and SA concentration. We do CFU counts. We also measure the concentrations of SPA within the sputum. And then we also recover the specific isolates of PA and SA that's involved in the infection using selective media, and then we throw it in an in vitro biofilm assay looking at PA antibiotic susceptibility in absent or in presence of SPA using the same SA isolate. So, and we also we're looking into doing whole genome sequencing with those isolates to further characterize. But for the purpose of this presentation, I'm going to show just the MyPack stuff for the visualization. So in terms of quantitative measures, there's so many ways you can measure through image processing. But to look at structural, I look at the biovolume of PA and SA, the number of aggregates, as well as the maximum aggregate size, which is just the largest aggregate in a single Z-stack image. And in terms of spatial analysis, I look at surface to biovolume ratio. So that's just the degree of aggregation. And then I look at the spatial distance of PA to the nearest SA. And because microscopy is the primary way I'm analyzing this data, my approach is very systematic. I take 3 biological and 6 technical replicates. We also use a 2-person imaging strategy. So myself and a technician in the lab named Shafanaz, we both take these images, and we just want to reduce image acquisition bias for that. And then we also scan in PA concentrated regions and capture those images, and then we go back in the same gel and then scan in SA concentrated regions to look at interaction from both perspectives. So I'll just share a set of 5 results. So the first result we wanted to look at is what is happening at the baseline sputum. So what's the effects of CFTR modulator therapy on PA and SA, and then what's the difference between pediatric and adult sputum. So looking at what's the effects of CFTR modulator therapy, so far with the data set we have, we're not seeing much of an effect, maybe a little bit less biovolume in the PA for pediatric, but for the adults, there seem to be no effects. So I have in the dark circles here are pediatric all sputums with CFTR modulator therapy, with the empty circles are the patients that are not on modulator therapy. But what we did see that is affecting the results is whether the sputum was collected from a pediatric patient or an adult patient. And for this case, the number of aggregates seemed to be greater in the adult patients compared to the pediatric, which was as expected. Now, in terms of staph biovolume, aggregate number and size and aggregation, it didn't matter whether they're on modulator therapy or not. We didn't see any difference, nor whether they were adult or a pediatric patient. So it didn't seem to have any effect on our results in terms of staph parameters. In terms of the difference between mono infection versus PA-SA co-infection, we did see an increase in the maximum aggregate size in the adult co-infected patients compared to the mono. There are also other things that are trending. For example, in the bottom right corner, there's PA aggregation. We're starting to see increasing aggregation in the co-infection in both the pediatric and adults compared to the mono. In terms of biovolume and aggregate size, this is only shown more in the adult patients where the co-infections has greater aggregate number compared to mono, and similar type of trend is happening with biovolume, but not significant yet. Although we did not see any difference between PA-SA co-infection compared to PA mono-infection in the pediatric patients, the amount of SPA concentration measured by ELISA seems to be positively correlated in the kid's sputum with PA biovolume. So this seems to be very important. And I think this is an interesting thing because, a lot of time when I'm visualizing these gels, in absence, if Staph aureus is eliminated, SPA is still present and it might be interacting more with Pseudomonas, so it might be something to look into more. In terms of the changes between time points, so looking first at the number of aggregates, one thing that that points out is from B to E in the pediatric patients, we do see a spike in the biovolume relationship, the number of aggregates, while in the kids, it doesn't, while in the adults, you don't see this initial increase from B to E. And then Staph aureus, for some reason, it doesn't change. It seems to be pretty steady no matter what time point it is at. And then when you look at the data collectively, we do see the same relationship where there's an increase from B to E time point. In terms of treatment with oral antibiotics, again, you see the same relationship. Staph is unchanged, not so much in the adults we don't see. And this is collectively, one thing is that oral antibiotics is very variable. So I guess depending on the antibiotics that's taken, so we don't see much of a relationship in that sense. The fourth results, looking at the maximum aggregate size of PA and SA, I find this really interesting is that when you look at the pediatric and adult patients, it kind of follows the same trend. So whenever B is increasing from B to E, SAs also seems to be increasing. So they kind of have sort of a relationship where they're both increasing at the same time. This is more evident when you look at the data as a whole. So you see from B to E time point, there's an increase in PA to PA as well as with SA for both the pediatric and the adult patients. Another interesting thing is about this maximum aggregate size. I find this parameter most interesting out of all is that despite normally you see the number of aggregates from the E to T time point drop, but this is not necessarily the case for the maximum aggregate size. So just maybe visually this will make sense. So here's an image from a adult patient at E day 0 and day 7 with IV antibiotics. You see that the number of aggregates is dropping, but then the maximum aggregate size remains, and it's about 21 microns for this particular patient. And this is a pattern that I've seen many times in the gels. Oral antibiotics, again, we see the same thing, that the maximum aggregate size does not necessarily drop between E and recovery. And this is the data as a whole. The last result is just looking at the spatial closenesses from the B to E time point. So this is just a normal distribution of a baseline sputum and then day 0 of a PEX. So you can see that there's planktonic cells with some PA and then by day 0 after baseline, you see that there's aggregates being formed. In MRS, what we do is we assign dots to the distribution. So the PA is assigned a red dot, whereas the SA is assigned gray dots, and then you remove the biovolume, and then you can measure PA to the closest SA to see the spatial difference. And by doing this, did this with 3 pediatric and 3 adult patients. This is a kind of a new analysis that we're looking at. And you can see at B, PA appears to be closer to SA, but at E day 0, one cluster of PA seems to be close to SA, and then it starts to move further apart because of these aggregates. So the spatial distance at PEX during PEX is changing. And this is just a visual representation of what I'm saying. So this is, if you're looking at the PA concentrated regions, you see that there's a lot of PA with some SA and SPA. And then the SA concentrated region, we see more interaction as well. But at day 0 of PEX, you see that they separate into their own niches or their own region of the same sputum, forming different aggregates. And then treatment, it kind of just gets mixed up and it's more random during treatment. So I don't really know how to analyze that part, but I do see a shift where they are forming separate aggregates. So to conclude, just the mono versus co-infection, PA-SA co-infection had greater maximum aggregate size in the adult sputum compared to PA mono-infection. And I did see a positive correlation between PA hydrogel biovolume and SPA concentration by ELISA in the pediatric sputum. So aggregate size and SPA might be some important parameters to look at. In terms of the changes between the time points, from B to E, PA aggregate number increased in pediatric sputum while SA remained relatively consistent. And also, there's a geospatial shift in PA distance to the nearest SA was observed in pediatric and adult sputum, where PA and SA became further apart and formed discretely large aggregates at PEX day 0 compared to their preceding baseline sputum. And then last, while PA and SA aggregate number decreased at time point T day 7 with IV antibiotics, the maximum aggregate size of PA remained unchanged in many of the adult samples and pediatric samples. So I just would like to acknowledge the lab from Dr. Waters, my supervisor, and as well as my co-supervisor, Dr. Yao, and everybody else in the lab, the research students who really help us to do all the in vitro work, and collaborators past and existing, also Dr. DePaz, who really helped me with the spatial analysis and the MyPack technique, the CF clinics, at Sick Kids St. Mike's as well as at McGill, and also just the participating patients and families in this study. Thanks. Thanks.
I'm curious about this really interesting phenotype of the bugs starting to separate over time. Do you think that's related to quorum sensing by staff and or this increase in SPA that staff?
Yeah, I do think it could be quorum sensing because it's, even though I'm seeing interaction at the baseline point, they are not physically interaction. I don't see much co-localization exactly. They're in the same field of view, but I don't see co-localization, which suggests that there might be some other form of interaction that's not physical.
While we're waiting for the next question, I have one for you. So I'm curious, I also like to kind of piggyback on that. I'm also curious about how during exacerbation, there's kind of a restructuring of the spatial arrangement of the Pseudomonas and staph. And I'm curious if you think whether that is driven by the host or by the microbes. And so, for example, Pseudomonas over time in CF will acquire inactivating mutations in the pili and the flagellum. So yeah, curious if you have any thoughts on that.
Yes, we are collecting data from the patients, each host, like different. So for example, that particular patient, they actually had virus. So that could be also causing the separation. I'm not too sure, but it could be the host as well.
Yeah,
So
yeah.
we're still we're gonna compare that data and see.
Okay,
Yeah.
super cool. Thank you. I'm sorry.
Congratulations.
Thanks.
So one short comment. So protein A, it's not supposed to be secreted. It's belonging to the M strands, microbial surface proteins recognizing adhesive matrix molecules. So in some strains, it's really secreted, but most of the time, it's on the cell wall of the bacteria attached.
Yes, I do agree with that. I see in the gels, I see most of the time it's membrane bound rather than secreted. Yeah. Cell wall, sorry. Yeah, cell wall bound. Yep.
And so my question is, so you're talking about oral therapy or also IV. So if it's related to Pseudomonas or also to staph or to both?
The oral antibiotics that's being taken from the patients. So yeah, so I guess it depends on how they're being treated. So you're right, there's variability in whether or not what type of antibiotics they are taking. And then that's why I see for the oral antibiotics, I'd see completely different trends, whereas it's more structured for the PECs treated with IV antibiotics. So I think that would definitely influence the results. Yeah, sorry.
Super beautiful talk. It's really interesting. I was really fascinated with the observation that the number of aggregates decreased during antibiotic treatment, but not the size.
Yeah.
I think many of us sort of expect that there will be a protection of the interior of aggregates from antibiotics, and so that result seems a little counterintuitive, and that's super fascinating and something that you can only get by this kind of methodology. I was wondering if you have any ideas about what might be driving this sort of aggregate heterogeneity in response.
Yeah, so I think that the antibiotics might not be being able to penetrate the large bacterial aggregates. So maybe that's why. And just the planktonic cell seems to be disappearing, whereas the large aggregates persist.
Well, that's a wrap on that. That was really cool. Very beautifully written. So I was just wondering, so in all of the restructure of the bugs that you see, how does that correlate, for example, with CFU and stuff like that? Like, for example, like in that image that you're showing right now,
Yep.
Are they number of bugs the same, and it's just that they are arranging them differently, and that's what maybe causing more symptoms, for example?
Yeah, so I do look at the correlations between CFU and PCR. On my poster, I have that. If I were to say what correlates best, CFU correlates best with what's visualized in the gels compared to something like PCR. It does not correlate at all. And I think that that's kind of used to guide, okay, whether I'm going to see something, but it doesn't correlate perfectly. Yeah, so.
That was so cool to get to see that data. I think we've all been seeking this actual signal where exacerbation is starting, and you have that in the increase in aggregate size. And I think I know I also always wondered if
if the Pseudomonas strains are swapping out and something new is emerging, and that's maybe why they're able to, maybe there's something new emerging on the strain level at the time of exacerbation. And I noticed you had NGS up there, so I was wondering if you could give us a sneak peek.
We have not done anything with NGS yet, but I think that's going to be something that's going to tell us mechanism or something more about why, like an explanation of why it's what's happening. Yes. So, thanks. Okay, yeah, that's something to consider. Thank you. Yeah, we do keep in that flow chart I was showing, we do have an extra vial of it. So we do have that preserved, and we would love to have any type of advice of what we can do with that to help explain. Yes, thanks.
One more, at least. Our lab group showed that Pseudomonas aggregates formation is promoted in the presence of dead neutrophils, but we were never able to actually replicate that with staph. Do you have any idea what the substrate or scaffold is for your Pseudomonas versus the staph aggregates are?
So the first part, so in terms of the neutrophils and...
Well, anything. Do you see more association with dead neutrophils with Pseudomonas and less with staph, and that's why they're separate, because they have different scaffolds?
I wish I had the image, but I do see a lot of neutrophils more with the Pseudomonas. I don't know if they're dead or active. So this is something that we are actually interested in exploring.
Thank you very much.
All right, next is Celie Gonzalez.
Good morning, everybody. Thanks to be here. Good morning, everybody. And thanks to be here. I'm really honored to share with you my research in cystic fibrosis today. My name is Celie Gonzalez. I'm postdoctoral research in Dr. Quinn Lab in Michigan State University. And today I'm going to talk about how this new triplet therapy that maybe you are familiar with this is changing the microenvironment effects in CF lung, and also how alters the microbial physiology, competition, and virulence. I have no disclosure for this talk today, but I have to say that I will show you some artworks by Dylan Mortimer in my presentation. In general, cystic fibrosis is an inherited genetic disease caused by a CFTR protein mutation, and this mutation causes defective mucociliary clearance in the airways, and also an abnormal accumulation of mucus in the airways. And this accumulation of mucus is a rich microenvironment in the airways, full of amino acid, mucin, and DNA that create a unique microenvironment very favorable for pathogen bacteria. The pathogen bacteria that is the most common in cystic fibrosis are Pseudomonas aeruginosa and Staphylococcus aureus, and these bacteria interact together in the cystic fibrosis lung to create polymicrobial infections. That's why the lung microbiome in cystic fibrosis is very different from a healthy lung microbiome. But nowadays, the revolutionary new therapy Trikafta improves CFTR function. What we know already from this new therapy is that you use 3 small molecules, tisacaftor and elisacaftor as a corrector, and ivacaftor as a potentiator. And what we know so far is that they bind to this defective CFTR protein and improve its function. This helps to the ion channel in the traffic to membrane, and improve the mucus in the cystic fibrosis airways. definitely the mucous physicochemical environment in cystic fibrosis is changing due to this therapy. And what we know so far from this new therapy is that decreasing the concentration of the main carbon source in this microenvironment. We know that because some groups has published that the metabolome is changing, decreasing the amino acid in the airways, and also the neutrophilic activity is decreasing, but we are still having persistent infection by pathogens. So now we are facing the Trikafta paradox, in which we already know how beneficial is this therapy for patients with cystic fibrosis in terms of lung function, reducing its sputum. But if the sputum is reduced, it's challenging for researchers to get samples and analyze or follow how the microbiome is changing in these new conditions. That's why we need new models and approach to study persistent infections. And we have designed an experimental model mimicking CF sputum pre-ETI and post-ETI in vitro to model the new microenvironments. And what we did was use a very well-known artificial sputum media that is full of carbon source, and we designed a new nutrient depletion artificial sputum media, decreasing each carbon source one by one, amino acid, mucin, and DNA. We use phenol red in this media as a pH indicator that tell us how the bacteria are changing their physiology. The synthetic, when we got this artificial sputum media in nutrient depletion settings, we built a synthetic bacterial community as a proxy for in vitro modeling of sputum communities. What we did was isolate the traditional pathogens in cystic fibrosis, Pseudomonas, Staphylococcus, Achromobacter, Stenotrophomonas, and other commensal bacteria. When we got this microbial community assembled, I incubate this microbial community in our artificial sputum media full of nutrient and with nutrient depletions in aerobic and anaerobic conditions, and after 48 hours, we analyze these samples for 16S, metagenomics, and metabolomics, generating data about its functionality, taxonomy, and metabolites. What we found as a few results was that the microbiome structure shifts under oxygenic conditions, but nutrient environment has little effects. And when we apply diversity statistical for this community, we found that in aerobic conditions, the diversity decreased, and in anaerobic condition, the diversity increased, as you can see here. But it's important to say here that this community is dominated by Pseudomonas aeruginosa, no matter the limited nutrient. However, when we look in deep about the physiology, we found that ETI mimicking nutrient depletions lower total low bacteria and alters nitrogen metabolism. As you can see here, the nutrient that most affect the total low bacteria in this synthetic bacterial community is when they are in artificial sputum media, low amino acid. And looking for these physiological changes, we measure the nitrates in this community grow in this different nutrient artificial sputum media. And also we found that in low amino acid, the concentration of nitrates, of production of nitrates in this experiment is driven by low amino acid. But knowing that nitrates is a metabolic strategy used for Pseudomonas aeruginosa, we decide to analyze these samples looking for small molecules, metabolites, as virulence factors produced by Pseudomonas aeruginosa. And what we found was that quinolones, rhamnolipids, and phenazines are decreasing in artificial sputum media with low amino acid. This is very interesting for us in this first experiment that is very controlled for the synthetic bacterial community nature. But how does this nutrient depletion in synthetic bacterial community can mimic what happened in fresh sputum communities? Well, we got 7 different fresh sputum from 7 different patients with cystic fibrosis. And we already know that microbiome in patients with cystic fibrosis is very heterogeneous. And it's important to say here that our P1 is a community dominated by Pseudomonas aeruginosa. As in synthetic bacterial community, we found that in sputum communities are also affected by oxygen, but highly variable depending on the individual. But again, when we look in deep in terms of physiology of this microbial community, we found that the artificial sputum media with low amino acid is changing the nitrix production in all of this community. Looking a little more in deep, using the metagenomic information, that we found that there is a high functional diversity in the sputum community under ASM, or artificial sputum media, but in low DNA. However, the community that is dominated by Pseudomonas is the community that present the more higher metabolic pathways. We analyze the metabolites for this community, focus on Pseudomonas aeruginosa virulence factors, and we found the same behavior in our synthetic bacterial community. Its virulence, Pseudomonas virulence, is decreasing in low amino acids. So, how do ETI, mimicking nutrient depletions, alters specific interactions between bacteria? We choose 4 of these main pathogens, Pseudomonas aeruginosa, and Staphylococcus aureus, Achromobacter xylosoxidans, and Stenotrochomonas maltophilia, and we perform basic microbial experiment of competition, but very informative in terms of functionality and physiology for us. We use artificial sputum media full of nutrients and artificial sputum media with low amino acid. And we culture these bacteria 1 against 1. What we found was that Pseudomonas aeruginosa is less competitive when grown in artificial sputum media, low amino acid, mimicking ETI conditions. Here, we already know that Pseudomonas aeruginosa has an antagonistic interaction with the Staphylococcus aureus, but in monoculture they grow very well. But when Pseudomonas grow together with in co-culture with the Staphylococcus aureus in artificial sputum media full of nutrients, Pseudomonas do its job. But when they are in low amino acid, Pseudomonas have the same growth rate, but Staphylococcus aureus can grow. That was very interesting for us because this interaction, this antagonistic interaction, is changing in terms of limited nutrients. What happened with Stenotrochomonas, we got the same pattern. In co-culture, when Pseudomonas grow in artificial sputum media mimicking the mucus pre-ETA, Staphylococcus aureus is almost vanished. But mimicking the mucus post-ETA, Stenotrochomonas can grow. What happened with Achromobacter? We found the same pattern. So this tells us that Pseudomonas aeruginosa, even in the community, is dominating. But in low nutrients, Pseudomonas aeruginosa is changing its physiology. So if Pseudomonas aeruginosa lowers virulence metabolites under low amino acid, may this impact host interactions? So we performed this experiment in collaboration with Dr. Sean-Pen Lee in Michigan State University. What they do was expose human lung epithelial cells to culture supernatant from Pseudomonas aeruginosa grow in artificial sputum media and grow in nutrient depletion artificial sputum media low amino acid. And they measure the transepithelial electric resistance. Here, we didn't see any significant change when we expose large airway epithelial cells to the supernatant. However, in a small airways exposed to Pseudomonas aeruginosa grow in artificial sputum media full of nutrients, we got a significant changes in terms of barrier integrity. So, as a conclusion for this serial of experiments, under nutrient depletion, mimicking ETI conditions, microbiome structure was not altered, but was altered by oxygenic conditions. However, in nutrient depletions, the microbial are changing its physiology and its metabolites. Also, in vitro, Pseudomonas aeruginosa is changing its production of virulence factors under low nutrients, specifically low amino acid, and these changes in Pseudomonas aeruginosa virulence factors could be an indirect evidence of the ETI effect in Pseudomonas aeruginosa and improving its virulence, improving for host cells, because Pseudomonas aeruginosa is reducing its virulence factors. So thanks to all these beautiful people in the Rob Quinn lab, and thanks to you for your attention, also to my family.
That was really nice. So I'm curious, I'm wondering why it was less of these virulence associated metabolites being produced, but then Pseudomonas is doing better. So I'm curious what the adaptation mechanism is doing, maybe aside from virulence in the host, but how is it impacting Pseudomonas' ability to survive?
Yeah, that's a good question. We think that Pseudomonas is one of the we know that Pseudomonas is one of the pathogens that are persisting infection in cystic fibrosis, even in people with under ETI. But the interesting things that we found here is that Pseudomonas aeruginosa is reducing its virulent factors as suggests less damage for the host because of the low nutrients. Yeah, it's the. Yeah, that's what. Yeah, thank you. That's what we try to do with co-infection with the Staphylococcus and Achromobacter anestino. Is reducing. It's like Pseudomonas is reducing its virulent factors against other bacteria just to try to stay alive in this microenvironment.
This is such a creative way of trying to understand this problem. And just to make sure I understand, so this environment, this post-ETI environment, would be the baseline health in people. And how do you anticipate that would change in people having exacerbations? Like, we've taken out DNA, amino acids, but do you think that during an exacerbation there might be more DNA because neutrophils are coming in, and like, is that something you can model?
Yeah, that's a very good point. No, we don't take in account the exacerbation, just trying to mimic how is the microenvironment in pre-ETI as an artificial sputum media normal formula that maybe some of you have used it. We just mimic how ETI is reducing the mucus, but no taking into account exacerbation. It's a good point. Thank you.
I was curious that so post-ETI you seem to have fewer free amino acids. So oxytrophic strains of Pseudomonas that depend on exogenous amino acids and how they compensate. I was wondering post-ETI if you have evidence that those strains are selected and that would apparently become less fit in this lower amino acid supplemented background.
Could you repeat the question?
If you lower amino acids, do you think there are certain strains that will completely drop out of their experiment because they need external amino acids to grow?
Yeah, one of the things that we know is that pseudomonas feed amino acids, and pseudomonas use other metabolites from other bacteria to feed and eat amino acids. But in this new condition, the nutrients are limited for everybody, like in microbial community. So it's, yeah.
I do have one little question. I was struck by the increase in the nitrates in the ASM condition. Do you think that has a role in controlling the ecology in the populations? It seems like that would be a bad thing.
Yeah. It seems like it's a bad metabolic strategy used mainly for pseudomonas when it's facing nutrient adverse microenvironment. So yeah, it's a kind of bad things, but at the same time, it's like the paradox that we showed. No, it's like ETI is improving, but also microorganisms are trying to stay in this microenvironment. But yeah, we still need more research on this.
Thank you very much.
Right. Thank you.
And our final speaker is Katrina Whiteson. She comes to us from UC Irvine, and she's a associate professor. And come on up.
All right, good morning. Happy Saturday. Thank you all for being here. I have the pleasure of sharing a really big data set with you that has been on my mind for more than, close to a decade, really. And so I'm really excited to share it with you, and I would appreciate any thoughts or comments on how to analyze this really valuable data set that was generated by many of the people in this room. I heard people refer to it as a cathedral yesterday, the STOP2 study, a medieval cathedral that took a lot to build. So I'm excited to hear what you think about the data. So I have no disclosures, and I want to acknowledge the team that made this all possible. I worked very closely with John LaPuma and Lindsay Caverly's groups at the University of Michigan, and some of the people in their labs. And I want to thank Sage Dunham, whose name is unfortunately blocked the way the slide turned out, excuse me, but he's giving a talk later this morning as well. And Jason Rothman, a postdoc who actually did a lot of the analysis I'm about to share. And here's some information about the posters and talks from our labs, and Sage's talk will be later this morning. So the way I look at the ecology of chronic airway infections is that you have these two major communities or populations. You've got densely colonizing oral microbes that have all these fermentative capacities, and then you have a more chronic opportunists emerging in the airways. You know, we don't have good if you don't have good immune clearance, then those bugs are not going to be able to be cleared from the lungs. And regardless of their proximity to one another, which I love to see, we're actually getting some biogeography data, like the MyPack data we saw earlier this session. But even if you don't know anything about the biogeography, we know that there's exchange of volatile compounds and other metabolites that can travel. And so that could be driving some of the altered microbial physiology and helping the chronic opportunists colonize, which actually has really been a theme emerging in this session I've enjoyed. So thinking about how I can come up with ideas for how we can tackle this type of problem, I consulted the Oracle, ChatGPT, and asked collective humanity what we could do to try to get some new tactics for tackling chronic infections in CF. And actually, the answer was surprisingly good, so it's motivating. Like, how are we going to do better than what ChatGPT could come up with? And anyway, a couple of the ideas are things I'll actually address in my talk. So looking at microbial community dynamics, looking at signatures, biomarkers, metabolites, maybe we can get some information that helps us understand the dynamics of the infection. So a couple background pieces of information that I think are really useful. I know everyone in this room has been aware of this paper for many years, as I have, but in 2012, John LaPuma's group published this really nice study that helped us see a trend that has stayed consistent through all the small studies we've been doing. And that is that as the infection continues, you often have one of the chronic opportunists, often Pseudomonas, coming to dominate the infection, and then as overall diversity is going down. So that's a theme we saw in a lot of smaller studies. And just to think about the chemical environment, we've just been hearing some really cool talks, even manipulating the chemical environment to understand the community. But the way I look at it, oxygen is often limiting, and essentially the airways are drowning in nutrients, often due to broken cells as a result of inflammation, and just a really an environment that's full of a lot of destructive molecules. So these factors are important to have in mind as you're looking at a big microbiome and metabolome data set. In some of the smaller studies in the last decade, we've started to see some metabolites associated with CF disease state. For example, when I was a postdoc in San Diego, I got to take breath samples from some of the CF patients in the clinic there. And we found butanedione, which is a fermentation product. It's often produced by Streptococcus, Rothia, or Staph. And we saw that going down with antibiotic treatment. And similarly, lactic acid, which has come up quite a few times this morning as well, is a molecule that has been shown to be potentially associated with the onset of exacerbation. So I've always had my eye on these molecules that are coming often from the oral microbes, which can travel and can start to become more abundant potentially. And they often have low pH, which is another indicator that could be really useful in following the infection dynamic. Each of these metabolites that we pick up has a story, and so it's actually, you can learn a lot by looking at metabolome data. Each of these molecules indicates either that a microbe has an active metabolism going on, or that there's a new condition, whether it's high nitrate or low oxygen. This will shift what kinds of molecules are being produced, both by the microbes and the host. So these are some of the molecules that have emerged in the last decade as I've been doing smaller studies. But now I've had this really amazing opportunity working with John LaPuma to look in a much bigger data set. And so this is from the STOP2 trial. And the study design is really an interesting and cool study design. I know many of the people in this room know more about it than I do, but the idea is that there's samples taken from people at the moment of the onset of an exacerbation. We don't know for sure whether whether antibiotics have been taken at that moment, but it's the moment that the exacerbation was diagnosed. Then people take whatever antibiotic they plan to take, so it's not controlled which antibiotic is being given. And then at visit 2, another sample is taken. And then at this point, it gets really sophisticated on the clinical side, but I am not considering that in my current analysis. I'm just bucketing all of the visit 3 samples in the same category. However, there were 2 groups, early responders or non-responders, and then in those 2 groups, they were each randomized to 2 periods of time for a continued antibiotic treatment. And the big conclusion in the 2021 paper from Goss et al. In the STOP2 study was that the shorter periods of antibiotic treatment were equivalent in efficacy as the longer periods of antibiotic treatment. So that's really interesting, but again, not the focus of my talk today. I'm just going to treat visit 3 as one sample. So we got 949 samples. That's a very big we. People from 60 clinics across the United States participated in this study. They come from 317 participants, and they are the same people. This subset of the study is when we got samples from all 3 samples from the same person. So we were able to follow people through time with this data set. And then we were very inclusive. We did exclude samples that had low sputum quality scores. Only sputum quality score 1 was excluded. And then anybody who had a very gross protocol violation was excluded. But we really tried to include as many samples as we could. This is kind of like just looking at the ecology of all the microbiomes and metabolomes of people with CF. in the United States, and during a very special time period, right? This is before many patients were taking the modulator drugs. So this moment will never happen again. It's a very interesting window into what the infections looked like before we had the modulator therapies. So I'm going to tell you about the two types of data that we generated. We did both 16S sequencing. This was carried out by the Lupuma lab up at the University of Michigan. I'm going to start there, and then I'm also going to tell you about our metabolomics later. So one thing that's interesting that emerges in this particular data set is that we don't actually see a big change in the total diversity of the samples through the visits. So here I'm showing you visits 1, 2, and 3, and the Shannon diversity. However, this also is very consistent with what we've seen in other studies, but most of the variance can be attributed to which individual donated the sample. So in this case, it's nearly half of the variance comes from the individual. There was no significance for disease stage or the visit on the 16S microbiome data set. The composition, again, is individual, and just to give you a sense of how they spread, an inordination plot, each of these points represents one sputum sample, and they're colored by disease stage, early, intermediate, or advanced. And then here's the disease stage diversity, and again, you can see there's not a big change in the Shannon diversity through disease stage on the microbiome data set. We do see big increases in Pseudomonas with disease stage, which again is very consistent with other smaller studies that have been going on in the last decade. And this also gives you a sense of the composition of the cohort of people in this study. So there's early, intermediate, and advanced samples represented, but biased towards intermediate and advanced. You can see some clusters emerging in the microbiome data. Two main clusters are emerging. If you look at how they are spaced on this particular ordination, they don't separate in a clean way, but you do see a difference in the diversity of the members of each cluster, and also cluster 2 in this case has a lot more people in it. If you look at the microbial composition inside the clusters, they separate really cleanly by Pseudomonas and Staphylococcus. And so this is consistent with so many of the things we've heard this morning, that Pseudomonas and Staphylococcus are the most abundant members of the bacterial communities in these infections. And also, interestingly, there's often cases where Pseudomonas is dominating and there's not as much staph, or vice versa. Streptococcus and Prevotella, this is all at the genus level, have a similar pattern to Staphylococcus with more abundance in the cluster 2, which also has a lot more people in it in this data set. And now I have another set of ordinations to give you a sense of how the Pseudomonas, how the different microbial abundances look in an ordination. This is an unsupervised, non-clustered, just looking at the samples view of the composition. So you can see that the samples dominated by Pseudomonas are really sticking to one side of the plot. And here with Staphylococcus, you see that they are often different than the samples that are dominated by Pseudomonas. I'm just going to go back and forth one more time because for me it's kind of nice to get to view this again. So there's the Pseudomonas abundance pattern, and then here's the Staphylococcus abundance pattern. So there are parts of this ordination where you see co-occurrence of Staphylococcus and Pseudomonas, right? But often they're separate. Streptococcus is more represented throughout the samples, so you don't see as clear of a clustering for Streptococcus compared to Staph and Pseudomonas. Okay, so that was my tour of the microbiome, and I welcome questions. Now I would like to get into the metabolomics. We used a semi-targeted approach. It's with a methanol extraction and a helic column, which kind of gives you the best of both worlds with some central metabolites and smaller molecules and also some larger polar molecules. So when you're choosing between GCMS and LCMS and you can't decide, well, I vote the helic column because you often can capture a broad diversity of metabolites this way. Okay, so just big picture, most of the metabolomic variation was explained by the individual in this study, even more than for the microbiome. We had 62% of the variance attributed to the individual. But we do have significant variance explained by both the visit time point and also the disease stage. So that's that did not happen with the microbiome data. So we're getting a different signal, maybe more indicators of visit and disease stage in the metabolomic data. Interestingly, we see this pattern that the metabolomic diversity increases at visit 2. To me, this may indicate that the antibiotics are busting open all the cells, and so we're seeing the contents of those cracked cells and the membrane bits reflected in the metabolome. It's interesting because when I was back in San Diego, and Rob Quinn is here in the audience, we had seen a signal at that time where we saw increases in metabolomic diversity with disease stage, which is the opposite of the pattern you see in the microbiome, right? We see microbiome diversity going down with disease stage. So it's interesting to think about that as the infection progresses. The load and diversity of molecules increases. And this is not the first time this signal has been seen. I saw Charles Esther present a similar result at NACFC almost 10 years ago, that you see a bigger density of molecules and a bigger diversity of molecules in samples from people with cystic fibrosis. So now we get to have the fun part of digging into what the significantly associated molecules are when it comes to visit. So here I'm showing you molecules that are significantly different between visit 2 and visit 1, or visit 3 and visit 1. And some of those molecules are familiar. In fact, there was a really interesting breath study in a pediatric research journal in 2010 where they found that the biggest signal separating children with CF from healthy children was essentially broken up cell membrane components, hydrocarbons of chain lengths that represented bits of cell membrane components that had broken up. And we see that same signal here associated with antibiotic treatment at visits 2, especially at visit 2. In this data set. You might notice some other familiar molecules in here, lactate, for example. We see a significant decrease in lactate at visit 2 after antibiotics. Lactate was the most abundant molecule in this data set. These are normalized relative abundance values that I'm showing you on the y-axis here. And that's quite significant that we see lactate going down after antibiotic treatment, maybe representing that the antibiotics are halting the metabolisms that are producing lactate, which seems helpful if the lactate is fueling Pseudomonas or other opportunistic pathogens. We also used a different approach for selecting the molecules that are different between the different time points. This is random forest, and we see the same lactate signal that I just mentioned to you. And another molecule that emerges is diacetylspermidine, which is an indicator of biofilms. It's actually used as a marker of colon cancer. And so that's actually quite interesting that we also see that that's going down at visit 2 after antibiotic treatment. If you look at metabolomic diversity with disease stage, there's not a hugely obvious trend, but there is significant variance being explained in our permanova with disease stage. So some of the variance in this data set is significantly associated with disease stage. If you cluster the metabolomes, you see 2 main clusters. Again, one is much bigger than the other, and the clustering for the microbiome is actually quite different. The people who fall into a cluster for the microbiome don't necessarily fall into the same cluster for the metabolome. But we see a spread in the diversity, a difference, less diversity in cluster 1, which is larger for the metabolome. And then if you look at some of those molecules that I was just talking about, both lactate and diacetylspermidine, you see differential abundances of those molecules in the 2 clusters as well. Now, here's a fun part to get to look at which of the significantly associated molecules are indicators of disease stage. So now I'm keeping the top 50 in here to give you guys a sense, and there's a lot of significantly different molecules between intermediate versus early or advanced versus early disease stage. Some of them also indicate destruction, so broken down nucleotides are some of the top signals. Amino acids are a really important signal that is consistently increasing with advanced disease stage. And actually getting back to the earlier question about, if you can remove the amino acids and how will this affect Pseudomonas growth, for example. I mean, there's just a lot of amino acids in the airways here. So I think that's an interesting challenge. I think it's oxygen limited and very nutrient dense. We're not the first at all to notice this amino acid enrichment in the context of worsened lung function. So here in a study from Deb Hogan, she has this nice analysis of BAL showing enriched amino acids associated with lower FEV1 status. So taking a deep dive into the amino acids, I'm now showing you the amino acid abundance across disease stage this is in order of significance. These are the amino acids that were showing up in our semi-targeted metabolomics. And you see this very consistent pattern that they are becoming more enriched with disease stage. So thank you for bearing with me on my tour of this enormous data set. There's so much we could do with this data. I really welcome ideas. Some of the big conclusions emerging are that there were subject-specific signatures explaining most of the variants on both the microbiome and metabolomic compositions. And we see indicators of antibiotic treatment efficacy emerging, and this could be useful. If you want to know if the treatment is working, seeing the lactate content go down, for example, could be a useful indicator that the antibiotics are working. We also see indicators of disease stage emerging. And so with that, I would like to thank my group and my collaborators who made this all possible. And I welcome questions. Thank you.
I was really fascinated with your observation that even though over time, as disease progresses and alpha diversity, so the microbial community decreases, you see increased metabolic diversity. And so certainly a big contributor to that could just be maybe the host response to infection. But I'm wondering also about intra-species genetic diversity over time and how, for example, later stages of infection, looking at Pseudomonas, we see high degree of mutational diversity within the Pseudomonas population, and could that also be associated with increased diversity of the metabolic landscape?
That's a great question. I don't know the answer to that question. That would be interesting to try in vitro to see if more diverse subpopulations have more diverse metabolic outputs. But I have to say, my feeling about where that diversity of metabolites is coming from, it's pretty clear. It's from like destruction. It's broken cell bits. It's like components of nucleotides. It's like broken down lipids that used to be part of the cell membrane. I don't know if it's host or microbial, to be honest. They could be broken up immune cells. They could be broken up bacterial cells. I think both. It's possible some of the molecules are specific to bacteria or host, but to be honest, many of them are just universal, part of biology type of molecules that would be hard to distinguish.
Gotcha. Thank you.
Yeah.
That was fascinating. I wondered if you were able to correlate or attempted to correlate any metabolomic signatures or features with or relate them to total bacterial load, either intact bacterial load or dead bacteria, or to host cells, like neutrophils, for example, or human genome copies or something.
Yeah, that is a very good idea. I have not tried to do that. I can tell you that the metabolome and microbiome data do not have great relationships. Like from a big picture perspective, a Mantel test yields nothing. There's not a lot of association. But I don't know if we have bacterial load data, but maybe I can find that out. John LaPuma might be in the room and know the answer, but or Lindsay. Thumbs down. Okay, thanks. But great question. The STOP2 data set is so rich, there might be other things we should try to correlate with. So,
Can I ask also, did you deplete extracellular DNA?
These were 16S, so no, we didn't need to. Yeah, which is, I'm a big fan of your host depletion method with the benzonase, but yeah, that's tough.
Hi, I'm Sam Durfee from Emory University. I was wondering, for the lipids that you found that were different between visit 1 and visit 2, can you identify whether those are host-derived lipids or microbial-derived lipids? Is that possible?
There might be some that are, and I don't know the answer myself right now. In fact, if there are people in the room who have knowledge of that, I think there are examples of lipids where I would be able to tell if it came from a microbe or not. But it's kind of early days for figuring that kind of stuff out. I think there are a few like well-known patterns that I should probably dig into to try to understand that better. So, but the answer is that I don't know. Yeah.
It is so exciting that, as you point out, it's a unique opportunity for sputum abundant. What do you know about the metabolites you saw? Are any of them volatile? Could we potentially, for lacking sputum these days, be able to take this data and extrapolate it to what samples we do have?
Like, for example, could we use breath sampling, which has volatile molecules in it, and learn from this data set? I mean, most of them all, this is a LCMS approach with a methanol extraction, so it's not as much focused on the volatiles. So from this data set, no, we actually have taken quite a few of these samples and also samples from another project with John LaPuma where we extracted the volatiles and analyzed those. My experience in comparison with breath samples is that even the volatiles from a very small volume of sputum are so much richer in molecules, like the same density of volatiles in breath, it just will be so much lower. It's very hard to get a big signal from breath in my experience. I know Heather Bean is in the room and might know more. But anyhow, I like that idea. That's a really cool idea, and I think we should be doing that more. So yeah, thank you.
Just quickly, the metabolomics data has like some surprising things, right? We don't necessarily think about when you see it, like drugs. Did you guys detect a lot of drugs, antibiotics, steroids in the samples, and how did you handle that?
The drugs did not land. So this is semi-targeted with 295 molecules that we did like a deep dive on quantifying. And then there's like endless additional molecules. So I totally hear you on that. The drugs were not among the top molecules. There was an artificial sweetener that was showing up actually. And having done a lot of other metabolomic studies, like for example, we have a 1,000-person saliva metabolome from people in rural poverty in an East Coast echo cohort. And in that study, we could see all kinds of stuff that like maybe that should be on the consent form. Like, do you want us to know about your use of various compounds? Or like maybe that facility, maybe the instrument was also being used for drug testing and it has nothing in a sample that was run before ours and it's not actually due to the individuals in that study. So I have seen that. And we also recently published a paper with John LaPuma where we quantified antibiotic content in sputum. And we did a really deep dive on that. We spiked antibiotics, all the most commonly used antibiotics for people with CF. We spiked them in. We did 2 different extraction protocols. And we wanted to know like where the antibiotics landing in actual people. And not surprisingly, our biggest finding would just be that the drugs with a longer half-life persist in the samples and are the easiest to detect. Yeah.
I'm going to return to lactate, and maybe naively, is there any correlation between the microbiome lactobacterial load and lactate levels? And do you actually see that in the microbiome?
I mean, that has been my favorite hypothesis for 10 years. So believe me, I got my eyes on that one. But to be honest, we don't see a clear association like that. And so then it reminds me as a biochemist that enzymes are processive and lactate production might not necessarily correlate with load of cells in that moment. I think it also has to do with flux in that moment and the molecules that are passing by. And if there's a bunch of glucose and not a lot of oxygen, you're going to get a burst of lactate. And I don't know how much that influences cell density. If it's low oxygen, everybody might be kind of scraping by and then they can just do a burst of lactate. I don't know. That's a really good question. It's not emerging easily out of compositional data from the microbiome in my experience.
And that's true even if you're looking at anaerobes in general?
It's really individual, and like John LaPuma and I had a really fun project, and some of our early samples, we had a few people who had big increases in anaerobes just before the onset of an exacerbation, and then we also saw increases in not just lactate, but many other fermentation products. So that was our main hypothesis going into some of these studies. But I mean, to be honest, the jury's out, like we're still figuring this out, but it's not a universal pattern in the data. It doesn't mean it's not happening in real life because these data are really obscured by host DNA and lots of other things, but it's not easy to collect longitudinal data from people and have a clear correlation between the microbiome and the metabolome. in my experience. Yeah, it's a cool question though. I thought we were going to see increases in fermentation products preceding exacerbations. And then I just got to see this beautiful data from Toronto where they were showing that the staph was actually decreasing in aggregate size and the Pseudomonas aggregates were increasing just before the exacerbation, which is contrary to my expectation. That's really interesting. So,
thanks everyone for joining us today. I hope you enjoyed our polymicrobial session.